When we're talking about this 3-person baby, we're starting to get into editing the germline.
So recently one of my friends asked me about the 3-person baby, and I don't know if you've heard about that in the news, but it's...
Whoa, I've heard it takes 2 to tango.
It does, but recently there's been a lot of media about the 3-person baby, and this is a baby that has DNA from 3 different people, which is kind of a crazy concept if you think about it, and that's something we're going to try and explain to you today. So you might remember from high school, I think it was in high school or middle school, where you learned about the mitochondria. And the mitochondria are the powerhouse of the cell. They're what makes your body work because they provide the energy that your muscles need, that your heart needs, basically everything in your body.
Right.
And so the mitochondria produce these energy molecules called ATP. And
ATP is always termed as the currency of the cell.
Yeah,
right?
Yeah, it is. So it's like the cell has to barter back and forth with how much ATP it's going to spend versus how much it can make from the mitochondria. And what's interesting about the mitochondria is the mitochondria has its own genome. It has 37 genes as part of its genome, and it's actually a circular genome, not the double helix like you're used to seeing.
So this is DNA that is outside of the nucleus?
Yes.
Okay.
Yeah, so it's still within the cell itself because the mitochondria is part of the cell, but it's not within the normal nucleus.
So what fraction of your DNA is actually in the mitochondria versus the nucleus?
Yeah, so there are 37 genes in the mitochondria. Our DNA normally has 20,000 genes. So that's about 0.18% of your DNA is mitochondrial DNA, which is a really small fraction if you think about it, like 37 versus 20,000.
So are the genes that are in the mitochondria, do they relate to the mitochondria function, I'm guessing?
Yeah,
okay.
Yeah, a lot of those genes are just involved in energy synthesis, and because ATP production is just so important. So the fact that the mitochondria has its own genome just shows that our cells need ATP so much. So getting back to the 3-person baby, so there are some diseases that are mitochondrial diseases. 2 of them are Leigh syndrome and Murph syndrome. These diseases are caused by a deficiency of ATP because the mitochondria DNA is mutated. So in Leigh syndrome, 20 to 25% of cases are caused by mutations in the mitochondria. And what happens when people have mutations in the mitochondria, they get a lot of seizures, they have some muscular dystrophy because the ATP is not able to get to their muscles, and so it's affecting things like their heart and their skeletal muscles.
Do they have effects in the brain? Because that's another tissue that kind of uses a lot of energy.
Yeah, they also have that as well. So sometimes they'll have some reduced mental and motor function, and that's one of the main issues with these mitochondrial diseases. But the interesting thing about the mitochondria is it is only passed on by the mother. So the father is not able to pass on mitochondria just because the sperm package is very small compared to the egg. So
it's basically just DNA, right?
Yeah, it's basically just DNA. So it doesn't have any room for mitochondria. So the issue with these sort of disorders is that if a mother suffers from a mitochondrial disorder, she's going to pass it on to her children, and she's going to pass it on to all of her children. And in some cases, a mother is fine to have this disorder and she can still function, but a lot of times if you have those initial mutations when you have a kid, those mutations will kind of build upon each other.
How can you be slightly affected? Can it be certain, because you have more than one mitochondria in your cell, so are certain ones affected but other ones aren't?
Yeah, that's exactly what happens.
Okay.
So it's this thing the doctors term heteroplasmy, and it's basically the ratio of diseased mitochondria to the ratio of healthy mitochondria.
Well, I imagine to a certain point, the healthy mitochondria in your cell can produce enough ATP for the cell to function, but beyond a certain threshold, you wouldn't have enough ATP and you would start to display these symptoms.
Yeah, definitely. And they've seen that in a lot of these diseases, the ATP synthesis is reduced by 50 to 70%. But the way that they measure someone's severity with this disease is by looking at the heteroplasmy count.
So how do you get around this issue of the mitochondria not functioning properly?
Yeah, so you make a 3-person baby. So what happens is you have the mother's cell with the diseased mitochondria, and you're able to take that nucleus out of that cell and put it in a healthy cell from another mother. So
wow.
Yeah.
So you extract the nucleus from the cell.
yeah, and then you fertilize it with the sperm. So it's kind of similar to in vitro fertilization in that way, but you get rid of the diseased mitochondria and you have healthy ones.
How are you able to take the DNA out of a cell? I mean, a cell is really tiny.
Yeah.
What, like 25 microns?
Yeah, it's really small. So they have to go and use these special microscopes and kind of have a little vacuum that like vacuums up the nucleus, and then they do some tests on the nucleus just to make sure that they didn't damage it, that there's no mutations, and then they're able to put this nucleus in the other cell. But it's a very delicate process, and one that has some controversy behind it as well. In 2016, a doctor from a U.S. fertility clinic actually went down to Mexico to perform this mitochondrial transfer for this family. So this mother had Ley syndrome, and she wanted to prevent her son from having this disease, so they did this mitochondrial transfer. And one thing they also did, just to make sure of the donor mitochondria, is they compared the mitochondrial DNA from the mother, from the son, and from the donor, just to see what mutations the son had, and comparing the two mothers' mitochondrial DNA just to make sure that there's nothing crazy going on before they put this in the son. And what happened is they were able to analyze this boy after he was born, and they've been continuing to follow his progress throughout life to see what his heteroplasmy counts are. So right after the boy was born, they did some tests just on different, did biopsies of different parts of his body just to see what the mitochondrial counts were.
How old is the boy now?
I think he's 3.
Okay.
Yeah. So they're still trying to continue to do tests on him just to see throughout time if there's anything that comes up.
Right.
So, because this is still illegal in the United States, which is why the doctor went to Mexico.
Yeah.
But
It's still kind of a new technique. I mean, we're getting to the point where babies that were made by in vitro fertilization are starting to become adults. So it'll be interesting, as the boy develops, to see if there are any issues related to the technique.
Yeah, I think it's definitely important too for researchers to be able to follow, so they can see, okay, because a lot of things in science, it's like we're really excited about it, but then it's like, how is this going to affect 10 years, 20 years down the road? So in this boy, they were able to see that in his circumcised foreskin, 9% of the mitochondria were affected and were diseased. So it just kind of goes to show that this technique, even though they were able to take out the mother's diseased mitochondria, there were some that still were there and still present. So this boy still has some diseased mitochondria, and hopefully that won't affect his ability to have a great life. But it's something to note that they're not able to take out everything. The defective mitochondria, they can still be present, but it would be much less than through natural conception. So, and interestingly, the UK approved the 3-person babies, this technique this year, for the MERS syndrome that I mentioned. So we'll be seeing probably a lot more about this, but as of now in the United States, this is not available for use.
When we're talking about this 3-person baby, we're starting to get into editing the germline, which is kind of something that scientists are cautious about dealing with. Anytime you're making a change to the germline, you're making a permanent change. It's going to be inherited. It's going to become all the cells in a new individual.
Yeah, and that's a hard thing because, so say you edit one child's DNA, that's going to be passed on to every single generation after.
Kind of gets into playing God, if you will.
It does, for sure. And with the mitochondria, it's even more significant because that is the energy producing molecule of the cell. So potentially, if you didn't realize the donor had given you mitochondria that had a disease in them or were prone to a certain disease, that could affect generations to come. And you may not have heard of mitochondria in the terms of ancestry as well. So they use mitochondria to trace back ancestry because the mitochondria is being passed on from generation to generation.
Right.
So it's a hard line because the future generations don't have consent, and that's one thing we talk about a lot as geneticists, is if you're going to do genetic editing, you have to have consent.
Right.
And a lot of people are willing to have it done to themselves, but then their future generations can't consent.
Right, and I think in some cases, like when we talk about these mitochondrial disorders that are pretty severe, people may be more likely to go to these extreme measures and use this kind of germline manipulation. But where do you draw the line? I mean, maybe most people would agree that to prevent disease you can use this technique, but what about beyond disease?
Yeah.
About enhancement?
Right. So it's hard with this sort of disease because these mothers want children, and they want children that are going to survive, because a lot of these children, if they're born with a mitochondrial disease, they'll live for 5 to 10 years and struggle through life for those 5 to 10 years. So this sort of treatment gives hope to those mothers, but it's also at what cost, and what is the future generation's role in this, because they're not able to consent to anything.
Right.
So it's just a way to think about if someone would do this and why, but also looking at other ways where there's kids to adopt or foster care, but
surrogate mothers.
Yeah.
Can you only get this technique done in the case of when you have mitochondrial disorders, or are you able to make a 3-person baby under other circumstances?
I think as of now you can only do it if you have this mitochondrial disorder, but I could see in the future them opening this up. But it is a very expensive process. I can't even imagine how much people are paying to get this done. It's similar to in vitro fertilization in the amount of cost, for sure.
And kind of risky. I mean, I'm sure they're not able to extract the nucleus perfectly every time. And they have to go through a number of eggs.
Yeah. So this 3-person baby, it's a really interesting concept. It's amazing that scientists can do this, but as always, with new science, there come ethical implications.
And the question, should we be doing this?
Right. So we're kind of here not to tell anyone what to do, but just to give people the tools.
To lay the facts out.
Yes, and show you what there is.